Hello and welcome back to AP Biology. My name is Dr. Tamara Philip, and in this lesson we'll be learning about cell membranes. First of all, we'll think about the cell membrane and how it's structured to form a fluid barrier between the inside and the outside of the cell. We'll learn about the model that we use for the cell membrane, which is called the fluid mosaic model. We'll also learn about large proteins that are distributed in the membrane, some of which go entirely through the membrane and do the job of transporting substances from one side of the membrane to the other. After we've considered that, we'll start thinking about internal cell membranes, that is, the membranes that compartmentalize different parts of the cell, such as the endoplasmic reticulum and the mitochondria.
The Global Teaching Project is doing a great job by giving segments of the difficult topics so that they can be understood in a much better way.
The Global Teaching Project, the thing that I really liked about it was that it kind of recreated a classroom.
With this age of globalization and intermingling, it is all the more necessary that all students everywhere have access to a good education. So the role that the Global Teaching Project is playing here is very, very admirable.
Welcome to AP Biology. I'm Dr. Tamara Philip, and in this lesson we'll be learning more about cell membranes and building on what we studied in the previous lesson. So before we had looked at the beautiful structure of the cell membrane and the complex structure of the cell membrane, which we describe as the fluid mosaic model, to take in the idea the membrane can shift around and that it also is studied in these beautiful proteins that make it look kind of like a mosaic. We talked about some of the simple functions of proteins in transport, in helping the cell signal with other cells, and even tethering the cells to a particular surface. And today we're going to look much more at transport. And we're going to be studying three different types of transport. We're going to look at passive transport, active transport, and then in the next segment we'll look at two types of transport called endo and exocytosis. Let's begin with passive transport. The passive in passive transport indicates that movement of particles across the cell membrane happens without an input of energy. So in passive transport, we don't require an energy source such as ATP. One example of passive transport is diffusion. In this diagram, we see an example of simple diffusion across a cell membrane. And in the left portion of the diagram, you'll see the particles are all concentrated on just one side of the cell membrane, which means that we have a concentration gradient. And as time moves on, we see the concentration gradient balances out until the third portion of the diagram, where we have equal numbers of particles on either side of the membrane. And so this kind of diffusion across cell membranes just happens without energy, because diffusion is a normal chemical event where things go from high to low concentration. The only thing to be aware of in diffusion is this can only work if the molecule is small enough to slip in between the phospholipids of the cell membrane. And the second thing is it only works for molecules which are polar, which can go in between the hydrophilic and hydrophobic parts of the lipid bilayer. What about particles that the cell might want to transport that aren't small and aren't polar? And there could be lots of different macromolecules that need to get into and out of the cell, and they can't just diffuse directly through the membrane. For them, where there's a process that we call facilitated diffusion. So it's still a kind of diffusion in the sense that the particles are going from high to low concentration, but it's facilitated by a protein that's embedded in the cell membrane. In facilitated diffusion, we have two kinds of proteins that we see at work, and the first protein that we'll talk about is called a channel protein. How a channel protein works is it provides a channel, a hydrophilic channel, through which particles can pass. And so it just allows far more particles to pass than could pass through simple diffusion. And a good example of a channel protein would be a protein called aquaporin. And as you might be able to guess from the name, it's something to do with water. So it's a pore for the diffusion of water. Let me try and draw a diagram of how it might look. In this aquaporin, we've got the cell membrane, and then the aquaporin is just stuck right through the membrane, and there's a hydrophilic channel that goes in the middle of this protein. Here's my phospholipid bilayer, or at least half the phospholipid bilayer, and then I've got my aquaporin protein going right through that bilayer. The reason that we need proteins like aquaporins, these channel proteins, is that diffusion is quite a slow process. And so if we have a channel protein positioned in the membrane, then it means that diffusion can just be a lot faster. And this is especially important in an organ of our body like the kidney, where the kidney is processing about 170 liters of water per day. And so it needs to have a lot of water diffusion going on. And the only way that it can sustain this quantity of water movement is through a specially formed channel. Okay, we're going to get up and we are going to pretend that we're proteins and phospholipids. And I want you to do a quick role play of how channel proteins work. Okay? So first you need to stand up, you need to talk to each other. How might you role play this?
Really, I'm not very strong. What do you mean? अब सब protein है, सब protein. नहीं, these two are the. Lauren, तुम यहाँ. Direction same होगी ना हमारी जाने. Going along the concentration variant, okay.
In addition to channel proteins, we have another class of protein called a carrier protein. Carrier proteins also perform a kind of facilitated diffusion, but whereas a channel protein is just open and allows the molecules or particles to go straight through, a carrier protein only opens when a substance engages with it. So in this diagram on the left-hand side, we see an example of this simple channel protein, and on the right, we've got 3 steps showing how the carrier protein works. In the first step, we see that the substance that is about to diffuse has gone into the protein and interacted with it in some way, and that causes a shape change, or what we call in biochemistry a conformational change, that changes the shape of the protein and allows the substance to diffuse the whole way through. The substance engages with the protein almost like a lock with a key, and that causes the protein to open up, and so now diffusion can take place. I'm going to have you stand up again, and this time I'm giving you only 2 minutes to figure out how you're going to demonstrate the carrier protein mechanism through a role play. Let's go.
To P for phospholipid.
Oh, we can do that, sure.
P for phospholipid. Yeah.
So if you can't go straight through, you have to cause that conformational change. Excellent.
No. We can get an extra minute in the next one.
In facilitated diffusion, it is still diffusion, so it still requires this concentration gradient, and the reason it doesn't need any energy at all, the reason it's a passive form, is that movement is driven by diffusion itself, right? The concentration gradient that we see across the membrane. However, there are also some cases in cells when we need particles to go against the concentration gradient. That is to say that we need them to go from an area of low concentration into an area of high concentration. For example, in signaling between nerves, we need to have things going against this concentration gradient, and the way that we can do this is through a form of transport called active transports. In this diagram, we're comparing passive and active transports. Passive transport would include simple diffusion straight through the membrane, or diffusion that is facilitated by a channel protein or a carrier protein. And none of these need energy, but active transport does because it involves going against the concentration gradient. And if you think back to our lessons on entropy and how the universe is going from order to disorder, when we're going against the concentration gradient, it actually goes against that second law of thermodynamics. It's going to order. And you might remember too that we said when we're trying to establish a situation that goes against entropy, we need an input of energy. So in active transport, we require not only a membrane protein, but we need ATP as well. There are some situations in cells where we have to go against the concentration gradient. For example, if we're trying to get sugar into a cell and out of the liquid surrounding the cell, that goes against the concentration gradient because typically cells contain sugar already, and so we need active transport to do that. And another example is maintaining the correct concentration of sodium and potassium ions that we need for nerve signaling. So we need to have a lot of sodium on the extracellular space, that is the space outside of the cell. And that won't happen naturally because it goes against the concentration gradient. So as you see in this picture, what does happen is ATP is required, so ATP binds to that protein, and that protein changes shape because of the ATP that's being used, and it allows these ions of sodium to go through into the extracellular space. So you're all pretending to be membranes and different proteins, and you're going to show these different mechanisms. So we've done channel proteins. We're going to try lots of different proteins. Let's go outside and try this out. So in this activity, you're going to pretend to be molecules in the membrane or phospholipids in the membrane. And I want you to do a role play of a channel protein. Now you're going to do the similar kind of thing, a role play, but this time with a carrier protein. Now we're going to take it up a level. You're going to do the active transport in a role play. So remember, in active transport, you also have to show the role of ATP. Good job with the role play. Let's go back up to class. So the only real difference really between active transport and a carrier protein in facilitated diffusion is the use of ATP. So in the diffusion, the particles go straight through the membrane or they diffuse straight through with the help of a carrier protein or a channel protein. And because those particles are moving down the concentration gradient, no energy is required. In situations where particles have to move against the concentration gradient, like when we're pumping sodium out of the cell, then ATP is required, and that helps create a conformational change in a protein that will allow that movement to happen. This is the end of the first segment of the lesson, and in the next segment we'll be learning about endo and exocytosis. Welcome back. Let's continue thinking about the ways that cells can move substances from one side of their membrane to another. In this part of the lesson, we will focus on two interesting and related processes called endocytosis and exocytosis. To get us thinking about these two different processes, I'm first going to show you a video in which we see a large irregularly shaped cell called a macrophage and two small dots, which are bacteria, surrounded by some different blood cells. And as the video plays, what I want you to keep an eye on is the behavior of the macrophage. And at the end of the video, the question that we're going to discuss is, what is the macrophage doing and how is it doing it? What is the macrophage doing and how is it doing it? Yes?
The macrophage is trying to engulf the bacteria. By moving, the bacteria is also moving. It must be having a tail-like structure which helps in its movement, and the macrophage is trying to engulf it inside.
Right. Okay, so let me paraphrase what you've said. The bacteria is moving around, and as you correctly say, it's probably moving with the help of a tail that is swishing from side to side. And the macrophage is chasing down that bacteria. And you use the word engulf, which we've heard before on this course when we talked about that theory of, in the beginning of cell development, a large cell engulfing smaller cells. So you're exactly right. The macrophage actually engulfs and then it goes on to digest the bacteria. And that's one of the ways in which our immune cells can protect us from infection. The process that macrophage is using is called endocytosis. So let's start talking about that one. And before we've broken words apart, let's do the same thing with this. So when it says endo, what does that suggest to you? Endo, an entry, yeah. And then cyto, we've seen before in the form of cell. So in endocytosis, what we're doing is bringing something into the cell. And the way that it happens is with big changes in the way that the cell membrane looks. In this diagram, let's imagine that this portion is the front edge of that macrophage that is chasing down the bacterium. What happens is the first thing in the engulfing, the macrophage starts to wrap a membraney arm around the bacteria, and it gets the bacterium into such a position that it's in a kind of impression or indentation in the cell membrane. And that little dip in the cell membrane changes shape so much that it's able to pinch off an entire circle or sphere of membrane. And the name that we give to that sphere of membrane that is now inside the cell is a vesicle. This is essentially how endocytosis happens. Bacterium or a molecule or something that needs to be internalized into the cell comes very close to the cell membrane. The cell membrane changes shape such that it forms a vesicle around that particle and pinches it off into a vesicle that is now inside the cell. Who could come up and do a diagram and a little explanation about how exocytosis might happen? So remember, endo is entry and exo is exit. So it's going to be almost the same mechanism. How would exocytosis work? Do you want to come and do that? Yeah, that would be great. Come.
So exocytosis would be quite opposite to what we saw in endocytosis. So the vesicle, which has a molecule here inside the cell, moves towards the membrane and finally moves out of the membrane.
Lovely. Yeah, very good. This is how exocytosis would happen. The cell would kind of package up something that it wants to release or excrete, and it would package it in this small membrane-bound capsule. What's the real word for this membrane-bound thing? Vesicle, yeah. So that vesicle travels to the cell membrane carrying whatever it's carrying, and then it fuses with the cell membrane, and then it kind of spits out or exocytoses the materials that it had been carrying to the cell membrane. Let's now think about what kind of things might be involved in endo and exocytosis. Can you start by doing a quick table sketch in your notebook of exo and endocytosis, and just jot down your ideas about what materials you think would be moved in endo and exocytosis. So we don't need a fancy table, just a quick T-chart. Just write down your ideas about what you think would be endo or exocytosed in the cell. What substances can we think of that undergo endo or exocytosis in cells? Charu, what did you write down there?
So for endocytosis, as we saw in the video as well, small bacteria and smaller cells which it can ingest, and also like the dead cells in our body which are eaten by macrophages. And for exocytosis, the waste product, whatever is there after digesting those bacterium or smaller cells, so that would be exocytosed.
Great, you're right. We've put together some of the main ideas already. Endocytosis can happen with small bacteria or other disease-causing organisms. If you think back to the video at the beginning of the lesson, we saw this immune cell called a macrophage, and it was sensing and chasing after that bacteria so that it could ingest it using the process of endocytosis. And that's one of the main ways in which our bodies are kept safe from infection, by macrophages traveling around our body, kind of sniffing out signs of infection and then going after and endocytosing those disease-causing particles. Endocytosis is really also important in dealing with dead and dying cells. If you think about the number of skin cells that we lose every day, we've also got cells inside our tissues that are dying, and those need to be cleaned up as well, and endocytosis would be one mechanism that would help with that too. In exocytosis, yes, cells are also secreting their waste products through exocytosis. And another thing that they do is they can secrete signaling molecules through exocytosis. This secretion of products through exocytosis is also another really important part of the immune response. Through endocytosis, cells also do this activity called pinocytosis, which literally means cellular drinking. And so what they do is they change the shape of their membrane to pull in small amounts of solutes from the extracellular space, and that's called pinocytosis or cellular drinking. So the pino part of that word connects to drinking, and this is also the root of the Hindi word to drink as well. So we get correlation between multiple different languages in this word root. And overall, as we think about substances being internalized and externalized from the cell, the thing that they all have in common is they're large things. They're things as large as a bacteria or a damaged cell, or like thousands of secretory molecules that would be released all at once. And for this reason, one of the words that we use to describe this kind of transport is bulk transport. Let's now think about the membranes that actually are inside the cell. This far, we've been thinking about the plasma membrane that separates the cell from the extracellular space. And to get you thinking about these membranes again, we're going to do another quick table and try and remember what we know. And in this, we're going to have 3 columns in our table. We'll say the name of the organelle, because remember, the membranes inside the cell are actually enclosing these things called organelles. Then we'll describe quickly the structure of the organelle and then the function. So I want you to copy this diagram, and I want you to do this for the nucleus, the mitochondria, and the chloroplast, which are the 3 organelles that we've studied so far. Let's hear about how we can start building up a picture of these membrane-bound organelles in the cell. Arzu, what did you put in your notes for the structure of the nucleus?
So it's an oval-shaped membrane with pores and DNA material inside it.
Yeah, it's a membrane. It contains these pores called the nuclear pores, and inside we've got the genetic material or the DNA. I don't think we're going to go through every single answer with question and response, but just to remind you, the function of the nucleus is to contain that genetic material and allow selected portions of it to exit for transcription or translation into proteins. And the structure of the mitochondria and the chloroplast, they both have these double membranes. And the mitochondrion is the site of cellular respiration. The chloroplast is the site of photosynthesis. Now that we've reviewed some of the functions and the structures of the nucleus, the mitochondria, and the chloroplast, we're going to learn about a new organelle for us, and that is called the endoplasmic reticulum. As you see in the slide, the endoplasmic reticulum is this series of folded up membranes that are just located outside of the nucleus. And the endoplasmic reticulum is important because it's the site of protein synthesis. The endoplasmic reticulum is located extremely close to the nucleus because the endoplasmic reticulum is making proteins using instructions that come from the nucleus. And so a piece of genetic material, which is required for protein synthesis, will exit the nucleus and it will go straight to the endoplasmic reticulum. In the diagram, you'll see that there are 2 sorts of endoplasmic reticulum. One of them is bumpy on the edges. It has these little black dots on the edges. And the other one is smooth on the edges. And these 2 types of endoplasmic reticulum are called, not very imaginatively, rough endoplasmic reticulum and smooth endoplasmic reticulum. And the roughness, those little black circles that we see on the endoplasmic reticulum, are caused by small structures called ribosomes. And the ribosomes are responsible for protein synthesis. The rough ER have ribosomes, and they're used to produce proteins, and the smooth endoplasmic reticulum have no ribosomes, so clearly they're not producing proteins, but what they do is they produce lipids and they carry out certain metabolic reactions. Now that we've briefly considered the endoplasmic reticulum, we'll move on to a different membrane-bound structure, and this is called the Golgi apparatus. And so the Golgi apparatus we'll add to our tables that show organelle and then structure and function. In this diagram, we see the Golgi apparatus, and in the graphic, the Golgi has been cut through so that we can see that it is really a series of flattened membrane discs that are kind of stacked up against each other. And you have to imagine that into the front of this stack, we've got things coming from the endoplasmic reticulum. So they might be proteins and they might be lipids. And they're carried there from the ER in those small membrane-bound vesicles. So you get a vesicle coming from the ER, it goes very close to the Golgi membrane, it fuses with the membrane, and now those lipids or those proteins are inside the Golgi complex. And what the Golgi complex does is it makes modifications to these molecules. So it might put a sugar on a lipid or a sugar on a protein to make those glycolipids and glycoproteins that we've talked about before. So the molecule progresses through the Golgi complex, and then eventually it will be transported to the outer cell membrane in a vesicle. And so even inside the cell, on these internal membranes, we see the processes of exo and endocytosis. Let's make a note of what we've learned about Golgi apparatus. And by the way, Golgi apparatus is sometimes referred to as the Golgi complex. The structure, we're going to describe it as a series of flattened discs, and there's a name for these, it's cisternae. And the function is the modification of lipids and proteins. A good analogy to help us think about the nucleus and the ER and the Golgi is to think about this process as being like a factory. So in a factory, you've got an office that gives instructions about what to make and how many and when. And in the cell, that would be analogous to the nucleus. And you also have a factory floor where people are busy putting things together. And in the case of the cell, that would be analogous to the endoplasmic reticulum. And then you might have a final processing center where people are making finishing touches to the product or painting or adding little bits. And that, in our model, would be equivalent to the Golgi apparatus. All of these internal membranes are very much working together and essential to the way that the cell behaves. And we've also seen that besides them making things, they have to transport materials as well. And in that transport process, they end up using the processes of endo and exocytosis, which we saw. This concludes the end of our lesson on cell membranes. And so just to take you right back to the beginning, we started off by considering the fact that cells need to move things in and out of themselves. One mechanism for this is diffusion. Facilitated diffusion by carrier proteins and channel proteins can really speed things up. And in cases where we have to go against the concentration gradient, for example, with the sodium and potassium pump, we need to input energy in the form of ATP. And then we looked inside the cell again, and we expanded our knowledge of organelles to include endoplasmic reticulum and the Golgi body, and we saw how the processes of endo and exocytosis are important in these internal membranes. Welcome back to AP Biology. My name is Dr. Tamara Philip, and in this AP Biology course, you're getting an opportunity to do an advanced college-level biology course for secondary students who have the aptitude and who have the work ethic needed to succeed at this kind of advanced level. This course, as well as giving you a rigorous introduction into biology, is also meant to prepare you to take the AP exam, or the Advanced Placement Exam in biology. That is the focus of this review session. AP exams are administered by an organization called the College Board. And the College Board has thousands of members representing US colleges and universities. And there are 38 subjects in AP, 9 in history and social science, 2 in English language, 8 in other languages, 7 in science, 5 in maths and computer science, 5 in the arts and 2 in research. And every year, over 4.7 million AP exams are taken, but most are clustered around the 8 most popular subjects. Biology is one of the most popular AP exams, and in fact, about 250,000 students are going to take the AP Biology exam this year. Why is it that AP courses are so important? So the first thing is that they can prepare you to be successful in college. Secondly, they can also help you to get into college, and they can help you to pay for college as well. Universities in more than 60 countries recognize AP exam scores in their admissions process. And most selective colleges look for applicants who've taken AP courses and done well in AP exams. Over 90% of 4-year colleges and universities in the US and in many other countries grant students credit and placement, sometimes both, on the basis of successful AP exam scores. AP tests are scored on a scale of 1 to 5, with 5 as the highest. Most colleges, but not all, give credit to students who score a 3 or above on the AP exams. That means that AP exams can help you graduate from a college in less time and at a lower cost. That's good. So how does the AP Biology exam work? This exam is 3 hours long, and it consists of 2 sections. So first you have a multiple choice section and then a free response or essay section. Both sections are 90 minutes long, and each comprises half of the grade. And the AP Biology exam is difficult, but most students do well on the test. And a substantial majority of secondary students who take the AP exam actually pass with a score of 3, 4, or 5. So what do you need to know to take the AP exam? The AP Biology exam tests understanding of 4 big ideas in biology. The first big idea is that the process of evolution drives the diversity and unity of life. The second big idea is that biological systems utilize free energy and the molecular building blocks of life in order to grow, reproduce, and maintain a dynamic homeostasis. The third big idea is that living systems store, retrieve, transmit, and respond to information essential for life. The fourth big idea is that biological systems interact, and these systems and their interactions possess complex properties. In this lesson, we learned about the movement of matter across membranes, which is relevant to each of these 4 big ideas. However, the AP exam focuses on how cell membranes relate to the second big idea. That is, how they help achieve a dynamic homeostasis so that concentrations of substances within and outside cells and organelles are regulated and maintained. The AP exam will test your knowledge of the following concepts related to the movement of matter across membranes. These are the concepts. Cell membranes separate the internal environment of the cell from the external environment. The specialized structure of the cell membrane described in the fluid mosaic model allows the cell to be selectively permeable, with the dynamic homeostasis maintained by constant movement of molecules across the membrane. Passive transport doesn't require input of metabolic energy or ATP because spontaneous movements of molecules occur down the concentration gradient from high to low concentrations. Examples of passive transport are simple diffusion and facilitated diffusion. Active transport requires metabolic energy and transport proteins to move concentrations against a concentration gradient across a membrane. Endocytosis and exocytosis move large molecules between the external environment and the internal environment. Eukaryotic cells also have internal cell membranes that partition the cell into specialized regions such as the nucleus or the mitochondria. And each membrane-bound organelle localizes reactions, including energy transformation in mitochondria, production or modification of proteins in rough endoplasmic reticulum, and the synthesis and packaging of molecules for transport in the Golgi apparatus. Now let's consider some sample multiple choice questions. The first question says, which is the most likely pathway that a secretory protein will take as it's synthesized in a cell? A. Plasma membrane to Golgi apparatus to ribosome to secretory vesicle to rough endoplasmic reticulum. B. Ribosome to Golgi apparatus to rough endoplasmic reticulum to secretory vesicle to plasma membrane. C. Plasma membrane to Golgi to ribosome to secretory vesicle to rough endoplasmic reticulum. Or D. Ribosome, rough endoplasmic reticulum, Golgi, secretory vesicle or plasma membrane. So we're thinking about the path that a secretory protein would take as it's synthesized in a cell. So we would therefore imagine that the first part of the path would involve the ribosome or the rough endoplasmic reticulum for synthesis, and that the final destination would involve exocytosis as the protein leaves the cell. So if I scan the different options, I can see that A and C are out of the question because, of course, the synthesis of a protein doesn't begin at the plasma membrane. So this leaves us with B and D. And if I look at B, it says it starts with the ribosome, which would be correct, and then goes to the Golgi and then to the rough endoplasmic reticulum. However, that makes no sense. So the answer would be D. The protein is synthesized at the ribosome on the rough endoplasmic reticulum. It then is transported to the Golgi and modified, packaged into a secretory vesicle, and then fuses with the plasma membrane. So the answer is D. Second question. The second question says that the movement of substances into the cell is dependent on the size, polarity, and concentration gradient of the substance. Which of the following represents an example of active transport of a substance into a cell? A. Diffusion of oxygen from the lungs into red blood cells in capillaries. B. Entry of sodium ions through a voltage-gated ion channel in a neuron cell during an action potential. C. The sodium-potassium pump, which restores resting membrane potentials using ATP. Or D. The osmosis of water into an epithelial cell lining the small intestine. If we consider the question again, the question is specifically asking for an example of active transport, which you may remember is the form of transport that requires ATP because it goes against a concentration gradient. Therefore, we can exclude answer A, because diffusion of oxygen from the lungs into red blood cells would actually be along the concentration gradient, and so passive. B is also not a possibility because it says that this protein channel or this membrane channel is voltage gated. So a change in voltage opens the channel, not ATP. And then C, the sodium-potassium pump, which restores membrane potential using ATP, is the most likely candidate because ATP is involved in active transport. And just to be sure, we can also exclude D because osmosis of water actually also goes along a concentration gradient without the aid of the energy source ATP. Question 3. An animal cell that is permeable to water but not salts has an internal sodium chloride concentration of 10%. If placed in fresh water, the cell will A. plasmolyze, B. swell and eventually lyse, C. endocytose water into a large central vacuole, or D. shrivel. So the answer to this is B, that the cell will swell and eventually lyse. So let me just explain why. So the cell is permeable to water, so water can rush in and out, and salts cannot. And then we place that cell into fresh water, even though the animal cell itself has a salt concentration of 10%. That will cause water to rush into the cell, causing it to swell, and then it will even lyse or burst because it's not being supported or resisted by a cell wall. Question 4. If plant cells are immersed in distilled water, they swell up as water moves into the cells. This process is called A. conduction, B. active transport, C. transpiration, D. osmosis, or E. facilitated diffusion. So this question is about the movement of water, and it happens when a plant cell is placed in distilled water, so when it's placed in a high water concentration environment, so the water will move due to osmosis, so that's option D. Question 5. A membrane protein opens to allow the passage of a substance from the intracellular to the extracellular environment when the intracellular concentration has reached a critical concentration. This protein is probably an example of A. a channel protein, B. a carrier protein, C. an ATP-driven pump, or D. an aquaporin. So if we look back at the question stem, we can get some more information about this protein. So it seems that membrane protein opens only at particular times, and that it will only open when an intracellular concentration has reached a certain level. So if it's only open at particular times, we can exclude option A because it's not a channel protein. And it could be B, a carrier protein, because these proteins work by a substance joining the protein or bonding with the protein and then causing it to change shape and then open. I don't think that C can be the answer because the question stem doesn't describe anything about energy or ATP. And D, aquaporin is just a distractor because an aquaporin is a kind of channel protein. And so this leaves us with answer B, carrier protein. Now let's try a free response question. So this is a kind of essay question, but it's been broken down for us. So the question begins like this. The relationship of structure to function is a critical idea in biology. A, describe the structure and nature of phospholipids and then explain how this relates to the structure and function of the cell membrane. And part B, describe the structure of the membrane proteins and then explain how their function in facilitated diffusion or active transport is related to their structure. And each portion of this question, A and B, is worth 4 points each. So let's consider part A. Describe the structure and nature of phospholipids and then explain how this relates to the structure and function of the cell membrane. So first of all, I would begin simply by describing what the structure and function of the cell membrane is, and then we would move on to explaining to how the structure and function of the cell membrane connects to phospholipids. So first, let's describe the structure and function of the cell membrane. First, a phospholipid bilayer comprises the cell membrane, and it contains integral or membrane-associated proteins. Secondly, some of these proteins extend right the way across the cell membrane, while others are only partially embedded in the membrane. Small molecules can move in between the phospholipids straight across the cell membrane, and also the cell membrane is the place where endocytosis and exocytosis take place. So that's how I would answer the first part of the question. I would begin by thinking about the structure and function of the cell membrane and then relating it to the exact chemical nature of the phospholipid bilayer. Let's look at part B. So part B, just to remind you, says, describe the structure of the membrane proteins and then explain how their function in facilitated diffusion or active transport is related to their structure. So now we're not looking at the phospholipids anymore, we're looking at those big proteins that are actually part of the cell membrane or going across the cell membrane. So first let's begin with a simple description of these proteins. I think since we've already described the phospholipid bilayer above, we won't mention it, but we'll talk about how these proteins are integral to the membrane. That is, they go all the way from the outside, the extracellular space to the intracellular space. We can talk about how a carrier protein might have a certain structure that allows it to bind a substance, and how a channel protein would have a hydrophilic channel running through the middle of it. And we can talk about how a protein involved in active transport would actually have a site where it can bind ATP. So having done a basic description, I think I would now move on to an explanation and begin by explaining that some solutes, like ions and larger hydrophobic molecules, can't cross the membrane directly, and so they need to go through a channel, so the hydrophilic channel of that channel protein. We can also explain about how in a carrier protein, the binding of a substance to that site that we mentioned above causes a conformational or shape change. And then thirdly, we can refer back to the fact that those proteins involved in active transport would have an ATP binding site, which allows them to undergo a conformational change and pass substances from one side of the membrane to the other.